Back in 1993, a biochemist named Michael Smith won the Nobel Prize for something that, well, it completely changed how we interact with biology. He figured out a way to precisely edit the code of life itself. And today, we're going to dive into exactly how he did that and how far we've come. So, what if you could edit DNA just like a document on your computer? I mean, literally open up the blueprint for a living thing, find a specific word, hit delete, and just type in a new one. That's the core idea we're exploring today. We're talking about making these incredibly precise, almost surgical changes to the genetic code. Okay, so the fancy scientific term for this whole process is site-directed mutagenesis, or SDM for short. And it's exactly what it sounds like, a method for creating specific, targeted changes in DNA. It's an incredibly powerful tool that lets scientists basically rewrite a gene's sequence on purpose. Why? Well, by changing the DNA instructions, they can see exactly how that change affects the final protein that gets built from those instructions. But why would we even want to do this? What's the point? Well, the applications are, frankly, mind-blowing. This isn't just lab bench stuff. SDM is absolutely crucial for understanding how proteins work, which in turn helps us unravel devastating diseases like Alzheimer's. It's a cornerstone for developing gene therapies for conditions you've probably heard of, like cystic fibrosis and sickle cell disease. And it goes even further. It helps us design new drugs and even improve enzymes that we use in all sorts of industrial processes. All right, so here's our game plan for this explainer. We're going to start at the beginning with the original M13 method. Then we'll look at how scientists made things way more efficient using plasmids. After that, we'll cover the PCR revolution, explore a really cool technique called DNA shuffling, and we'll finish up by looking at how we can literally expand the alphabet of life itself. It's going to be a fun ride. Okay, first up, the original M13 method. You know, every great invention has a version 1.0, right? The first draft. And for gene editing, that was the M13 method. Think of it as the foundational proof of concept. It was a little clunky, for sure, but it proved that this incredible idea was actually possible. So the process actually started with a virus called M13. What scientists would do is create a tiny synthetic piece of DNA, we call it a primer, that was almost a perfect match for the gene they wanted to edit. The key word there is almost. They would intentionally build in a single tiny typo. You can think of this primer as a bookmark you stick in a book, but this bookmark has a spelling mistake written on it. The cell's own natural DNA copying machinery comes along, sees this bookmark, and uses it as a starting point to copy the entire strand, faithfully writing that typo right into the new genetic code. And you can see exactly how that typo works right here. That long strand is the original DNA from the M13 virus. The shorter piece is our synthetic primer, and you can see it binding right on. But look closely at that red letter. It's a G that's paired with an A. That's the mismatch, the intentional error. And here's the cool part. The cell's copying machine, the DNA polymerase, it just doesn't notice this tiny mistake. It comes along and copies the entire circle of DNA using our primer as the template, which permanently locks in that change. So what you end up with is a mix of viruses, some with the original unedited gene and some with your new mutated version. So the big question is, how well did this actually work? I mean, in a perfect world, you'd expect a 50-50 split, right? Half original, half mutated. But in reality, it was bad. The success rate was just 1%. Yeah, you heard that right. Only about 1% of the new viruses actually had the mutation. That meant trying to find the one that worked was like searching for a needle in a massive haystack. So yes, the technique worked, but it was way too inefficient to be practical for most scientists. Okay, so clearly a 1% success rate just wasn't going to work. Scientists desperately needed a better way. And the solution? It came from ditching the viruses and instead using these small circular pieces of DNA called plasmids. This next chapter is all about cracking that efficiency problem. And the difference between the old way and the new plasmid method, well, it was like night and day. The M13 method was super complex. It needed all these extra cloning steps and even required special strains of E. coli just to get it to work at all. But the plasmid method? So much simpler. A much more direct workflow. It worked with just your standard off-the-shelf host cells, and, this is the big one, it was way more efficient. And here's the really clever part. The genius of the plasmid method was using multiple primers at the same time. So one primer would create the mutation you wanted in your target gene, just like before, but, and this is the key, a second primer would simultaneously change a different gene on that same plasmid. one that gives the bacteria resistance to an antibiotic. So what does that mean? It means only the bacteria that successfully picked up the mutated plasmid could survive when you dose them with that antibiotic. It was a brilliant built-in filter to automatically get rid of all the original, unchanged plasmids. And the result of this clever trick? The efficiency skyrocketed from that measly 1% all the way up to around 90%. Now, look, this wasn't just about a bigger number. This was a complete game changer. It was the difference between an experiment taking months of painstakingly screening colonies and getting your answer in just a few days. It transformed mutagenesis from this really difficult, specialized technique into a reliable, everyday tool that thousands of labs could use. The pace of discovery just exploded. All right, but the evolution didn't stop there. The next huge leap forward came with a technology you've probably heard of, the polymerase chain reaction, or PCR. So if plasmids made this whole process efficient, PCR made it lightning fast, super cheap, and incredibly flexible. It really opened up a whole new world of what was possible. You can think of this as a menu of the different genetic edits you can now order up, thanks to PCR. By designing your primers in clever ways, you could do so much more than just change one single letter. You could do a point mutation, sure, swapping one base for another. But you could also do a deletion, completely removing a chunk of a gene. Or you could do insertions, adding in new bits of genetic code, both small and large. The flexibility was just amazing. But what if you don't know exactly which change you need to make to get the protein you want? Well, PCR has a clever answer for that too. It's a technique called error-prone PCR. And again, it's exactly what it sounds like. Scientists intentionally mess with the conditions of the PCR reaction to make the DNA copying enzyme, well, sloppier. They make it more likely to make mistakes. This sprinkles random typos all over the gene, creating this massive library of different mutants. Then, you can screen that whole library to find a version that has a cool new property, like an enzyme that can suddenly withstand much higher temperatures. Okay, so we've talked about making super precise edits, and we've talked about making a bunch of random mistakes. But what if you wanted to get even more creative? That's where a technique called DNA shuffling comes into play. A great way to think about this is it's less like editing a sentence and more like being a DJ. You take a couple of different songs, chop them up into little pieces, and then remix them to create something totally new. And this visual explains the concept perfectly. On the one hand, you have random mutagenesis, what we just talked about. That's like adding a few random typos. You see them there as the red stars. DNA shuffling is totally different. It takes whole chunks, entire sections, from related genes and swaps them around. The goal is to create these new hybrid proteins that hopefully combine the best features of both of their parents. So how do you actually do this? Well, you start with a set of related genes, maybe from a few different species. You use an enzyme to chop them all up into random fragments. Then, you throw all of those fragments together into a PCR machine. Now, because the original genes were related, the fragments have similar sections, and they start sticking to each other, acting like primers for one another. And the DNA polymerase just comes in and stitches them all back together, creating full-length genes that are now completely new, shuffled-up hybrids. Now, everything we've talked about so far, it all operates within the normal rules of nature, right? It uses the same 20 standard amino acids that life has been using for billions of years. But this final technique, this is where things get really wild. This is about pushing past those limitations. It's about expanding the very genetic alphabet to build proteins with totally new chemical powers. And this diagram shows the absolutely brilliant trick they use to do it. Okay, look at the left. That's the normal process. The cell's machinery reads the genetic code, it hits a stop signal, a codon like UAG, and protein production just stops. You get a short, incomplete protein. But now look at the right. In a specially engineered E. coli cell, scientists have added some new parts. They've essentially taught the cell to read that same UAG stop signal not as stop, but as insert this cool new thing here. And that cool new thing is a brand new synthetic amino acid, that blue circle. This lets them build a full-length protein that now contains a building block that nature has never, ever used before. And look, the crucial thing to remember is that this isn't just some fascinating science experiment. These tools have profound, real-world consequences. This quote here really drives it home. It says that by using these exact methods, scientists have been able to dissect how protein plaques form in Alzheimer's disease. They're understanding the fundamental mechanisms, and that is what paves the way for future treatments. So when you step back and look at the whole journey, it's incredible. We went from a clunky method that only worked 1% of the time to being able to literally rewrite the building blocks of proteins with things nature never invented. This whole field has given humanity an unprecedented power to edit biology. We've truly gone from just reading the code of life to actively writing it. And that power is immense, which leaves us with one final big question to think about. We can now write new words into the code of life. What stories should we tell?